Okay. Hello everybody. Good evening. Actually, strength is very less. I think it is quite understandable. Most probably you all are having your exams, right? CSIR. So, any of you are writing? Okay. So, without wasting much time, so let's start. Okay. I will not take much of your time. So, I think screen is visible to you. Alright. Okay. So basically we are going to discuss today about the neuronal inhibition. So you must all are aware of the neuronal inhibitions and the related questions that you must have studied in your there is one topic, right? Neuromuscular junctions and all in the syllabus. Okay, so we'll basically deal with that from openly, and we'll see to make it more easier. Okay, so there are different types of inhibitions. Okay, so in terms of neurological inhibitions, we'll find, see, there are different kind of inhibition, right? Presynaptic inhibition, postsynaptic inhibitions, and you will find there are other inhibitions like feedback inhibitions and okay. Screen is not clear. Okay, guys, actually one thing you can do, you can access the slide from the link. Okay, because I have already shared the PPT there, so you all are must have the access to get this PPT, right? So you can even download and see in your computer, because it might be a condition like sometimes network and all fluctuates, so the screen quality might be poor. Okay, so you can just go to that link and open your PPT. Okay guys, let me discuss. So we were discussing different kind of inhibitions, right? So there are different kinds of inhibitions like feedback inhibitions. Inhibition can be, your voice is not clear, so can you take the earphone? Sure, why not? Now is it audible? You can just take it out. Okay. Okay, so I was talking about different I was talking about different kind of inhibitions. Okay, so you must be aware of feedback inhibitions. Here we are discussing today the presynaptic and postsynaptic inhibitions. So this is one important aspect in terms of the complexity in the inhibition process, right? So I would like to make it more clear. See, presynaptic. Okay, so we all know that what is synapse. Synapse is the point where the dendrites of a neuron is connected with the axon of the previous neuron, right? So that particular area is called your synapse. Now, if it is presynaptic inhibition, that means before the synapse, the inhibition is taking place. Okay. Now, let me clear what are the conditions how the inhibition can be taken place. So there are two ways mainly. Okay, the first one is that secretion of inhibitory neurotransmitters. Okay, so what are the inhibitory neurotransmitters? For example, GABA. Okay, now what they do, we have already discussed. They they opened up the negatively regulated ion channels. Okay, like for example calcium ion channels. Okay, mind it, these questions might help you in your examinations, many examinations. Okay, all the inhibitory neurotransmitters open up the negative channels. Okay, negative ion channels. Okay, uh, and all the positive one or excitatory neurotransmitter opens up the positive ion channels, that is sodium ion channels, all that. Okay, so I told you that presynaptic inhibitions taken place is before the synapse, the point number one. Second point is that it is basically done by two ways. One is your by inhibition through neurotransmitter, and which one is the neurotransmitter? Generally GABA or like that, which inhibits the which open up the negatively charged ion channels. Okay, for example chloride ion channels. Second point, how it can be done? By GPCR signaling. Okay, so these are the two methods by which the presynaptic not termination takes place. Okay, now for example, if it is done before, if it is stopped before the synapses, as you can see, the downstream signaling or downstream proliferation of the signals will decrease. Okay, this is by presynaptic. Now one more condition can be done, is it is the postsynaptic. See, presynaptic termination always taken place in the synapse or before the synapse. Okay, it is during the transmission of stimulus. Okay, that is basically regulated by your neurotransmitters, and these neurotransmitters are basically regulated, for example GABA, during presynaptic not termination or not signaling. Okay, now postsynaptic. Postsynaptics means after the synapse. Or what is after the synapse? If it passed the synapse, you will find the dendrites leads to the cell body. Okay, and further it will lead to action, right? Now, if that is the condition, you will find here two terms, right? IPSP and EPSP. Okay, what is that? Excitatory potential, okay, and inhibitory potentials. Now, how these what is basically these two? Okay, so excitatory जो होता है, EPSP जो होता है, ठीक है, it excites, right? So that means it's positive, it's open of the positively regulated ion channels, for example sodium channels and all. Similarly, in IPSP जो है, it will regulate the inhibitory channels. Now the resulting signals, okay, resulting signals, whether it is excitatory or inhibitory, will be decided by both of these. If EPSV is more, then the signals will transverse. However, if it is neutralized, for here in this figure you can see it is neutralized. Okay, one excitatory and one inhibitory neuron fires simultaneously. So the resulting results will be blocked at that particular points. Okay, so the nerve stimulus will not further proliferate, and you can see postsynaptic can block all the entire neurons. It will blocks all the way to transfer the stimuli. However, in presynaptic, since it is before the synapse, you will find at some particular point, it's basically depends on the neurotransmitter. So suppose there are 5 vesicles and you will find you block 4, but still you can find 1 or 2 can transfer the stimuli, and that will result in the transmission of signals. But in postsynaptic, it is after the synapse, okay, because it is not regulated by neurotransmitter. It is basically regulated by the stimulus. Okay, so if the potential is not at all generating because of the polarization level, see, EPSP is because of depolarization, right, and IPSP is because of hyperpolarization. You all know, right? That is, hyperpolarization is the maximum opening of potassium channels that will release in the hyperpolarization. Okay, so if hyperpolarization will be there, that will be balanced by your depolarization, and there will be the resulting stimulus will be 0 or even less than that. So there will be no stimulus that will proliferate. But in presynaptic, it is basically depend on the neurotransmitters, and neurotransmitters cannot block completely. Okay, so if even the neurotransmitter will be blocked, or suppose inhibitory neurotransmitter gets started, you will find some amount of signals can transfer, as you can see in this figure. Okay, So basically we are going to deal with this because you will find there are three questions in here, that which of these follow, these are basically examples. I cannot discuss about the individual molecular mechanism of all these toxins, right? So that's why I supposed to discuss about what is the mechanism and why it is occur, right? So which of this is a synaptic blocker at neuromuscular junction? This is something like physostigmine, okay, and tetrodotoxin is your neuromuscular junction, it is the presynaptic blocker, and bungarotoxin is your postsynaptic blocker. So you can mark it off for your NPTEL final exams. Okay, you'll find the examples, few examples as well along with this, that the tetrodotoxin is your presynaptic blocker, bungarotoxin is your postsynaptic blocker, and synaptic blocker at neuromuscular junction is which of these is a synaptic blocker at neuromuscular junction, that is your physostigmine. Okay, so these are basically theoretical questions, you can just find it out. Okay, rather you don't need to go into the details of this particular composition of, because these all are toxins. Okay, now the next question is that a product from Clostridium botulinum, okay, that is your purple sulfur bacteria, you must aware of, right? Prevents fusion of Dax vesicles with postsynaptic membrane and prevent the neurotransmitter in the synapse. Now, if you can see the figure 1, you can see what is the mechanism. Okay, let me discuss how it is occur. It is basically occurred because the neurotransmitters is found in the vesicle, we all know, right? Now, you must aware of this snare protein, right? Okay, Surya, I'm reading the question is that which the Clostridium botulinum जो होता है, Clostridium botulinum, so basically you need to understand how it is affecting which neurotransmitter and how it is affected. Okay, this is the question. Okay, so now the point is that the normal neurotransmitter जब release होता है, when it is released, it is formed in a vesicle. Okay, and if you have read the vesicular transport, you will find there is a snare protein. Okay, there are even snare 1, 2, something, there are names, 25, A, B, C and all. So there are snare proteins. These what this basically done, these proteins, there is a snare proteins in the vesicle and there is a snare protein in the membrane. There are different subunits that I can see. Okay, now all these are compatible. Okay, so the vesicle, the protein associated with the vesicle bind to the protein associated in the membrane. As you can see here, see in the below, the membrane area, you will find in the synapse there are snare proteins. Okay, so when snare will fuse, basically there are different subunits. Okay, when they fuse, the vesicle get destroyed and the neurotransmitter get released. Okay, so if the snare protein is degraded, that is what the in botulinum toxin do. Okay, so the botulinum toxin, what it does, it enters into the vesicle, it degrades the snare protein as well as from the membrane. Okay, if there is no snare protein, the vesicle cannot bind to the membrane, and if it cannot bind to the membrane, it is not going to fuse. Okay, because it is also a disorder if it is fused just randomly, right? In order to fuse, it has to bind to the membrane. Okay, and then in order to bind to the membrane, it needs to have the snare protein. Okay, so if it doesn't have the snare protein, as in case of the toxin, botulinum toxin does, what it does, it breaks down the snare proteins, and the breakage of snare protein is not it doesn't make it compatible to bind to the membrane, and that's why the acetylcholine vesicle is not going to fuse. And if it is not going to fuse, you will find it will not going to release. Okay, this is very crystal clear. And if it is not going to release, the nerve signal is not going to transmit, because acetylcholine is a excitatory neurotransmitter, right? So this is what the concept behind this question is. Now if we'll find the next question, the next question is that which of this is not a constituent of olfactory system? Okay, so if you have seen, the olfaction is a very important aspect of our sense, right? So there are different systems, there are different areas. Olfactory bulb, that is basically presence in the brain, in the frontal brain, right? Or you can say, okay. Then we have all, yes. Okay, screen is not clear. See, what you can do, you guys, I can just suggest you guys, you can find the slide in the, I have already uploaded it. The link that is accessed to you guys, you can just find the slide, you can just download and you can see. That I can tell. Even from the next class, you can do that, okay? Before coming to the class, you can just go and download the slides so that you can just listen to me. Okay, because actually I can understand, because I am not in my lab, so I'm a bit skeptic about the network and all, because yeah, it can be possible that the screen is might blurry. Okay, but what I can do for you, I can just stop sharing and share it again. Now it is fine. Okay. Okay. Okay, so now we are here, right? So just a minute. Okay, so we are here that we are discussing about the olfactory system, right? So if you will find the olfactory bulb, that is a part of our nervous system, olfactory epithelium, that is a part of your epithelial system. Okay, which question? Yeah, we are in the 5th question only, right? Okay, so which of this is not a constituent of our olfactory system? Okay, so in the olfactory system, you will find the olfactory bulb, that is a part of our nervous system, and this olfactory epithelium is part of our skeletal system. So in the olfactory bulb, there are several reasons. There are several cells that they have different functional specificity that we can discuss, but when we will discuss about this particular system in details. But here the question is that which of this is not a constituent of the olfactory system? Okay, so if you see, this is the very specific cell, okay, mitral cell or top cell, that is only found in olfactory bulb, or most probably found in olfactory bulb. Okay, apart from this, you will find Bowman's gland, that is the one is responsible for the secretion of your mucus, right? And your mucous layer, it is the major area of our olfactory system that protects it from external dust, allergies, microbes, all these things, right? So all these things are part of your olfactory system. However, stapes is present in your ear. Okay, so that is a part of your not a part of your olfactory system, rather a part of your ear. Okay, so this, if you want, we can discuss this structure in detail some days. I will take some class on this. Now, which of these secretes mucus into the surface of olfactory membrane? Okay, so this question is that, as I have already told, right, this is the Bowman's gland. Bowman's gland, also called as olfactory gland, okay, is one of the branched tubulovascular glands situated at the mucous membrane of the olfactory region of the nasal cavity. These glands produces mucus to moisten the olfactory epithelium and dissolve odor containing gases. Okay, so Bowman's gland is basically responsible for the secretion of mucus. Okay, so these are the theoretical questions whether I should ask you, but there is no, I have already highlighted the answers, right? So which substance is mixed in natural gas because even low concentration can be smelled? See, मिथाइल मैरकैप्टन विल बी द आंसर, एंड मिथाइल मैरकैप्टन का कुछ स्पेसिफिसिटी है दैट मिथाइल मैरकैप्टन जो है इट इज ऑल्सो कॉल्ड मिथाइन थायल. Okay, thiol means because of it has sulfur group, and mind it, whenever there is sulfur you will find the rotten tomato or rotten egg smell, you know. So methanol is, that's why it is basically what is the property of it? It can be act as an odorant, it can be act as a flamboyant, that means it can easily catch fires. Okay, that's why it is mixed to the natural gas, that basically LPG is used in our home. That's why the smell in the LPG is because of this particular chemicals, that is methyl mercaptan. Okay, so just remember these questions you will might face in your exams in NPTEL. Okay, if you are going to supposed to give examination for this. Okay, and you should give, because you will get certificates and all, right? And it will help you in further if you are applying for something, you can just put it in your CV that I have NPTEL in this, that. Okay, so now 1 decibel represents an actual increase of how many times? So I will just tell you that 1 decibel sound is basically 26% of your normal sound energy. Okay, if somebody asks you or somebody tell you that it is 1 decibel increase, or I have got 1 decibel increase in the this or that, so you will find that is 26%. So here you will find the answer is 1.26. Why? Now the thing is that suppose I will give you 1 decibel sound, 1 decibel sound is there, and you need to, what is the, how many times actually increasing it is. Okay, so basically it is 26%, right? So if it is 100, then you will find 126. Okay, so if it is 1, you will find 1.26. Okay, so these are again the question. Now the 9th question is that the length of the basal fiber in ear beginning at the oval window going from the basis of cochlea to the apex. See, this particular things, that is your basal fiber, is very interesting one. It is started from the oval window. Okay, if my cursor in the slide is visible, it is starting from here, it will go till the apex, right? Here it is the apex, it will go till the apex, and again it will come back. So this length is increasing. Okay, because it is starting point. Oval window is the point where the basal membrane starts, and it will go till round window, that is here, you will find it here. Okay, round window. So the journey of this basal membrane, basal fiber, starts from the oval window to the round window. So it is starting, so it will get increasing. Okay, it increases. So the next question is that when sound becomes louder, the amplitude of vibration of basilar membranes and hair cells, what? See, if you can see the slide in here. So this is your basilar membrane. Okay, now when there is a sound, if you can see this figure, here you can see, suppose there is a vibration. Okay, the vibration from this side. Okay, eardrum, you can see here eardrum, right? So in eardrum, whenever there is a sound, okay, there will be a vibration. Now this vibration will impact you this because see, you can see the bone is associated with the oval window, right? Now whenever there is a sound is generating, okay, whenever there is a sound is generating, the vibration is going to vibrate this bone, that is basically stapes here, right? This is basically stapes, malleus, incus and stapes, right? So whenever there is a vibration, whenever there is a noise, the vibration will vibrate the stapes, and the stapes is directly associated with your oval window. Now this oval window is going to vibrate, and as I told you, from the oval window your basilar membrane is started. Now if you can see this figure, the concept is clear, right? Whenever there is a noise, the bones will get vibrate, and the bones is associated with your oval window, and if oval window gets vibrate, here in this figure it is more clear that from the oval window, the basilar membrane starts, right? Now this basilar membrane is associated with this particular hair cells. Okay, so when the basilar membrane vibrates, this hair cells will vibrate, right? And if the hair cells will vibrate, these cells are associated with the auditory nerves. Okay, so once these cells vibrate, the nerves will get what we can say, the stimulus, they will get the stimulus, okay, because of the vibration, and further that will get transmitted to the auditory nerve, because these all are basically part of these are auditory nerves only. Okay, so that is basically associated with the cochlear nucleus, right? Cochlear nucleus is the point, or in the cochlear nucleus all the auditory nerves are get gathered. Okay, so once the basilar membranes vibrate, the hair cells vibrate, and the vibration of hair cells will vibrate the nerve axons, and these nerve axons get stimulated and they will further transmit the signals. There are different nerves, you can see 1 kilohertz, 4 kilohertz, 16 kilohertz, that will get stimulated, and further that will transfer the nerves, or transfers the stimulus or signals or neurons, you can say, through these auditory nerves to the cochlear nucleus. And this is the mechanism by which this noise in terms of stimulus comes to our brain, and we'll be able to define the noise, we'll be able to identify the noise, and we'll be able to sense the noise. Okay, so this ends with the today's slides, because there are majority of the slides are mainly theoretical questions are there. You can just find only here is the concept is that you can find it will go, and you all should remember this structure. Any of you can draw a structure of ear at any point of time. Okay, structure of ear, structure of eyes, because these are the very basic questions that you can face in daily life. Okay, anybody can ask you, and what is the mechanism, how it is affecting, right? So this is very simple, that this is one-way traffic. The noise generates the vibration will vibrate the window, and the window will vibrate the basilar membrane contains hair cells, and the hair cells vibration will generate vibration or stimulation for your nerves that will sensed by our brain, right? So this question is done, and this is basically 1 decibel increases 26%, that is 26% means 1.26, 126, 1260, whatever. Okay, now the next question is that methyl mercaptan is basically used for odorant, and it has very smell, and that's why it is found in natural gases. Bowman's gland is responsible for your mucous secretions. Stapes is found in ear. Okay, as I told you, this is a part of malleus, incus, and stapes is a part of bone system. 3 bones found in the ear, right? And this is the mechanism you find that this botulinum toxins cleave the snare protein, and that's why it is not being able to bind to the membrane. And if it will not bind to the membrane, the vesicle is not going to release out. The vesicle is not going to burst. Okay, if it is not going to burst, it will not going to release acetylcholine, and it will block your neuronal transmissions. Okay, not signaling. Similarly, these are 3 toxins. You need to remember this. We don't have other options. Bungarotoxin is your postsynaptic blocker. Tetrodotoxin is your presynaptic blocker, and physostigmine is your synaptic blocker. Okay. Now we discuss about the presynaptic inhibitions, where it is before the synapse by 2 ways. That is, one is your by blocking your by GABA, that is your inhibitory neurotransmitters, or based by your GPCR signaling, that is basically blocked. And the second one is your postsynaptic inhibition, that is basically done by the stimulus, that is basically IPSP and EPSP. So it is depend on the hyperpolarization and depolarization balance that will decide whether the stimulus will be there and will be transmitted or not. Okay, so that's it about today's class. If you have any doubt, you can just come across to tell me so that we can discuss. Else, Else, whoever of you are going for examination, all the best to all you guys. And let's see. Okay, see you in the next class then. Till then, thank you and goodbye. If you, okay, 10th question. Okay. Yeah, 10th question is that this is the, as I have discussed, right? See, this is the mechanism, okay? Whenever a noise is generated, here, this is your, if my crosshair is visible, this is your eardrum, right? So this tympanic membrane or eardrum is going to vibrate. Once it will vibrate, here is your malleus, incus and stapes. These bones are going to vibrate. If this is vibrate, this is associated with your oval window, right? This oval window is again vibrate. Now, from the oval window, your basilar membrane is starting, right? Now, see this image. Here, this is the image of your basilar membrane. This is the image of your basilar membrane. Now, if this is the image of, this is the basilar membrane, see, here you can see the oval window, right? Now, oval window is vibrating means the basilar membrane will vibrate, and the basilar membrane is associated with the hair cells. Okay, so if the basilar membrane is vibrate, the hair cells will vibrate, and the hair cells are associated with your neurons. Okay, now if the hair cells are vibrating, the neurons are getting excited, they will get the stimulus. Okay, if they will get the stimulus, then the neuronal signaling part will take place. Till this is the mechanical part that is occurring in the ear. Okay, that this is all this transmission of vibration, right? From the generation of noise, the vibration will transfer through bones, through tympanic membrane, through the basilar membrane, and through hair cells. So the vibration that is started at the point of tympanic membrane will vibrate these hair cells, and this vibration will start or stimulate this nerve stimulus or nerve endings that will transfer the signals, that will transfer the neuronal stimulus. Okay, so this is what about this question is. So this is basically all associated, so that this vibration will generate the stimulus, and that will get transported, or that stimulus is forwarded till the cochlear nucleus, where the nucleus have a bunch of nerve endings that is confined to that particular position, that is further is going to your brain to sense the auditory reflex, you can say, right? Okay. Okay. Okay, 8th question is that देखो 1 decibel क्या होता है, ठीक है? What is 1 decibel? Okay, so 1 decibel is actually increasing in the sound energy, that is 26% energy. You can read the sentence. 1 decibel change in the sound. Okay, if 1 decibel change in the sound equates to about 26% difference in sound energy. Okay, question is about energy, mind this. It is not about sound, it's not about unit. We are not talking about decibel, how many hertz or all these things. We are not talking. We are talking that if I am asking that 1 decibel change in a sound or 1 decibel change in sound, okay, that means 26% change in the sound energy. Our point of focus is the energy. Okay, so 1 decibel changes in 26% changes in sound energy. Okay, now the thing is that if it is 26% change in energy, now suppose let's think we have 1 decibel change. Okay, that means how many and we have 1 decibel sound, then till what it will change? Okay, it will increase 26%, like that is 1 plus 0.26, because 26% of 1 is 0.26, right? So it is 1.26. So 1 decibel represents actual increase of 1.26 of sound energy. If I will ask, if we will talk like 100, we have 100 decibel, then what will be the change in the sound energy? 126. Okay, so we are talking about that 1 decibel change means 26% change. Okay, that's it. Now it's based on the unit, and you all are masters in manipulating the units, right? So this is the point: is the 1 decibel change means 26% in the change in energy. Now this question is like, if there is nothing mentioned, that means it is starting from 1. If it is 1, then it will 1.26. If it is 100, then it is 126. Matlab, how many times changing? Okay, it is 26% changing, right? So if it is 100, then it is 26% changing. That means it will 126. If it is 1, it will 1.26. That's it. Okay. Okay, guys. Then see you then. Thank you all for attending the class. Thank you so much. So this is 8 weeks. So you will have 4 more weeks, and then you will have your examinations and all. So till then, start preparing beforehand. If you find any doubt from any topic, even I might not be able to explain, but we'll get back to you as soon as possible. You can mail, and you can get it clear. Okay, it's not about to getting the certificates, it's about to getting the concept clear so that you can take the help of this course in future. Okay, then thank you so much. See you in the next class.